In this session, we'll learn how to use a program called SeqBuilder to evaluate DNA sequence features and identify restriction enzyme sites. We will be using a commercial bioinformatics application suite by DNASTAR. They have provided a free educational license available for teaching purposes. An important concept for this session is DNA restriction enzymes. These are enzymes that cut DNA at specific patterns called palindromes. You may be familiar with other palindromes like the words race car, civic, and kayak, which are the same in both directions. Restriction enzymes that recognize rare sites are very useful for a variety of purposes, while enzymes that target common sites generate a lot of small fragments that are difficult to resolve. Restriction enzymes are initially isolated from bacteria, and their names typically reflect the bacteria of origin. For example, EcoR1 is from E. coli. This picture shows the DNA sequence targeted by EcoR1. After digestion, the DNA fragments shown below are the resulting products of that digestion process. Let's use the SeqBuilder application within DNASTAR to take a closer look at the CFTR gene that we downloaded in an earlier exercise. So we'll launch SeqBuilder, and then we'll find the file for the CFTR gene that we downloaded in GenBank format. When we first open the file, we notice that there are U's presented. This is because the default is to show an RNA sequence. If we go to the sequence menu and change RNA to DNA, we see the more appropriate version of the representation. Now let's go to the Edit menu and choose Select All to select the entire sequence, and then go to the Sequence view and choose Statistics. This provides information including the GC ratio of this DNA sequence. So now let's learn more about the EcoR1 sites in the CFTR gene. Choose Enzyme Manager under this Enzymes menu and scroll down to EcoR1, and we can see the sequence pattern that was shown in the earlier slide. Now let's learn how to identify the EcoR1 sites in CFTR. We expand all enzymes alphabetical to the group that contains the E's, find EcoR1 and select it. And then if we scroll down in the sequence, we'll see that it displays the sites in the sequence that are targeted by EcoR1 as shown here. When we want to understand how many total restriction targets are in a gene, you can select all sequences, and then it shows every possible site for every known restriction enzyme. But as you can see, that can provide an overwhelming amount of information. You can filter this in a number of ways, including by choosing only those enzymes that targeted sequence patterns of 6 or more nucleotides. This generates a much more manageable list. In this session, we've learned how to identify some DNA sequence patterns called restriction enzyme targets, as well as how to identify statistics about a long DNA sequence.